guys, I wanted to quickly go over a couple of things with you for cell transport, just to make sure that we're all on the same page about some of the key terms that we're learning about, okay? So one of the words we've been talking about is concentration gradient. And that simply means that there's a difference in concentration, okay? So if you have a beaker and say it has a divider in the middle, and you have some sort of molecules on both sides, okay? But there's a difference, right? There's not the same number of molecules on this side as there is on this side. So that means there's a concentration gradient. This is in low concentration, this is in high concentration because there's more of them, okay? So why this is important is because all of the things in cell transport either go with the gradient, meaning naturally from high to low, or they go against the gradient where they go from low to high, okay? Now you can kind of remember this by thinking about a hill, okay? So say we have this hill, right? And you're at the top of it. And you want to roll down the hill. Is that going to take a lot of energy? Not really, because you just kind of make yourself straight and roll down, right? And you don't have to do anything. The force of gravity is just rolling you down and it takes no energy. But imagine you're at the bottom of this hill and you got to walk up, okay? Is it going to take energy to walk up to the top of that hill? It sure is. So going from high to low requires no energy. It's like rolling down a hill, and going from low to high needs energy. Okay, it's like walking up the hill, okay? Now, in this scenario, we call this passive transport. Passive transport. This, because it's requiring energy, this is active. Okay, and there's a couple different types of passive transport, and they were diffusion and osmosis. Now, diffusion is anything moving from its high to low concentration gradient without the help of anything else. Okay, there's also something called facilitated diffusion. And facilitated diffusion is still molecules going from high to low, but in this case, it's also being helped by a protein. Okay, so we're going to say protein helper. Okay, so facilitated diffusion is just diffusion, but with a channel in the protein. And what that means is that when you have the membrane, some things can go through it and some things can't. Well, in this case, facilitated diffusion is helping larger molecules or charged molecules get through the membrane by creating a hole in it, basically, that they can go through. They still don't need energy because it's just a pore, meaning it just stays open and things can just go, it's like an open door and things can go through it, okay? Now, osmosis, this is where we're talking about water moving. Okay, so anytime we're talking about water moving from high to low, we're talking about osmosis, okay? But all of these are going from high to low concentration. All of them require no energy, okay? So then over here on this side where we have active transport, we have a couple of different things here as well. We have endocytosis and we have exocytosis. Okay, now this endo sounds like enter. That means in, right? To enter a room, you go into the room. So endocytosis means that things are going into the cell. Exocytosis sounds like exit. And to exit, it means out. So exocytosis means things are going out of the cell, okay? So these are the big ones as far as what is happening. You can also talk a little bit about pinocytosis and phagocytosis here when we're talking about endocytosis. So we have pino and phagocytosis. Pinocytosis, we sometimes call that cell drinking, because it's when the cell is taking in really, really small particles or liquids, mostly liquids, okay? So that's why they say, call it cell drinking, pinocytosis, cell drinking. I kind of remember this. You might not know what Pinot Noir is, but it's kind of a wine. You drink wine, Pinot, you know, Pinot is grapes, so cell drinking. Phagocytosis is cell eating, because it is larger molecules in things and nutrients that the cells need, okay? So it's larger pieces of the cell. So we have active, then we have endo and exo, meaning going in and out of the cell. Going into the cell then is pinocytosis, which is cell drinking, and phagocytosis, which is cell eating, okay? So, okay, in other words too, I wanted to mention, people were confused about if it went against or with a gradient. So with gradient or against. Okay, and what that means is that, is it going with its natural gradient from high to low? So with would be high to low, and against would be low to high. Okay, so it's like against what it normally would do naturally, and with is what it would do normally. So high to low, low to high. Okay, so hopefully that helps clarify some things, and maybe you can add those to your notes and make adjustments to that cell concept map that you put in your books. All right, take care, guys.